The cell cycle. There are two main stages, the growth stage and division stage. The growth stage, or interphase, can be broken down into three phases: G1 phase, S phase, and G2 phase. The G means growth, and is often called gap. The S means synthesis. Cells generally sit at G1. That's why they work, doing the things they do. It's called growth phase here because it immediately follows division, and there's rapid growth so the cell can achieve its performance potential. When the cell needs to divide, and depending on the cell depends on how often this occurs, the cell moves to S phase, where the DNA needs to be replicated so that the daughter cells following division have the same complement of DNA. Gap 2 sees the cell increasing in size and developing the extra organelles and proteins, etc., to ready itself for mitosis, the phase that sees the nucleus dissolve, DNA organize itself into two identical groups, and new nuclear membranes forming around each DNA group, and cytokinesis, the cell actually splitting in two. Interphase in three phases, the bulk of the cell cycle. Generally, more metabolic activity continues throughout this stage. Example, if the cell is supposed to produce a hormone, then that's what it does during interphase. During gap 1, the cell grows following division and performs its normal metabolic activity. During the synthesis phase, DNA is replicated. The specifics of this will be discussed in another chapter. Gap 2, the cell grows to ready itself for division. The cell synthesizes more organelles, proteins, etc. to facilitate the division and make sure the daughter cells have a good start in life. In mitosis, DNA separates into two identical groups. It's the shortest part of the cell cycle. Not all cells divide very often. Muscle cells and nerve cells divide very infrequently. Typically, you have all the brain cells you're ever going to have at childhood. Cells that die off into adulthood and old age are generally not replaced. So if anyone tells you you have a brain of a child, take it as a compliment. Cells that divide more frequently are the cells that become blood cells, cells that line the digestive tract, and skin cells, for example. During mitosis, the cell splits into two, a process actually called cytokinesis. There are specific stages to mitosis that will be discussed next. Why do we have mitosis? Well, it's required for growth, maintenance, repair, and reproduction for some organisms. Some organisms don't enjoy sexual reproduction to propagate their species, and they rely on asexual reproduction. The phases of mitosis are prophase, metaphase, anaphase, and telophase, and you must remember these, and in this order. During prophase, the DNA supercoils from chromatin to sister chromatids, or chromosomes. The nuclear membrane dissolves, allowing the chromosomes to take up a whole cell. The centrioles, in animal cells only, separate to opposite poles of the cell, where one end of the microtubule strings, called spindle fibers, anchor. The other end of the fibers anchor to the centromere of the chromosomes. So each chromosome is connected by two spindle fibers, one anchored at each pole of the cell. It's worth noting that there are several types of spindle fibers, and each with different names: astral, kinetochore, and polar microtubules, that contribute to manipulating the positions of the chromosomes and assisting in cell division. It's not necessary to distinguish between them in this course. Also, I tend to oscillate between the term chromosome and sister chromatids. The two are the same thing, because by convention, the presence of a centromere is the definition of a chromosome. However, your book occasionally, and so do I, calls a chromatid a chromosome. It's not worth getting hung up over this, because it seems to be unimportant in Biology 30. Metaphase is the longest phase of mitosis and sees the chromosomes align along the equatorial plate of the cell, guided by the spindle fibers. The cell does not move to the next phase until all the chromosomes are in a row. If a karyotype has to be done, this is the preferred phase of cell division to observe for chromosomal abnormalities. Anaphase sees the contraction of the spindle fibers, which cause the separation of the sister chromatids to each pole of the cell. Telophase and cytokinesis complete the mitosis events, ensuring the identical groups of chromosomes are at opposite poles of the cell. A nuclear membrane forms around each mass as the chromatids unwind and form chromatin again, and the cell undergoes cytokinesis. So to summarize, in prophase, the chromatin supercoils to become chromatids connected at the centromere with their identical chromatid. The structure would become then sister chromatids, or a chromosome. During prophase, the nuclear membrane dissolves. The centrioles separate, and the spindle fibers form. One end anchors to the centriole, the other to the centromere. Each chromosome is connected by at least 2 spindle fibers, anchored at each pole. During metaphase, the spindle fibers direct the chromosomes to the equatorial plate. It's the longest phase of mitosis, and karyotypes are preferred at this phase. Anaphase. The spindle fibers contract, causing the sister chromatids to separate to opposite poles of the cell. And finally, telophase. A nuclear membrane forms around each set of chromatids. The chromatids begin to unwind and form chromatin, and cytokinesis occurs. The biological clock. It turns out cells can't divide forever. It seems a biological clock dictates the number of cell divisions a cell can endure. A heart cell cultured in a petri dish can undergo exactly 50 divisions. Even if the cell is frozen after 20 divisions, it will divide another 30 times once it's thawed before dying. 50 divisions, no matter how long the freezing or what stage the cell division was suspended. The biological clock is turned on at the point the zygote undergoes differentiation. Therefore, to say mitosis produces exact copies of the mother cell might not be entirely accurate. In fact, evidence has shown that shortening of the telomeres, the terminal ends of chromosomes, become more pronounced following every mitotic division. This suggests a mechanism for aging. As certain cells begin to reach the end of their dividing capacity, symptoms associated with aging occur. Graying hair, increase in facial lines, atrophy of body processes and systems. There are 2 cell types, however, that seem to escape the confines of the biological clock and continue dividing rapidly and frequently. The cells that escape aging are the sperm-producing cells, upward of 1 billion a day from puberty until the day he dies. And cancer cells. Cells that cannot regulate their rate of division are cancer cells. They divide before they're mature and ultimately form a mass of non-functional cells called a tumor. They do not mature to develop the control mechanisms to halt division before they divide again. If cancer goes into remission, it's believed it's because the surrounding non-cancerous cells have successfully transferred chemical intercellular messages to the cancer tissue to trigger the tumor suppression genes, which inhibit the oncogenes, genes that cause cell division.